Okay guys, now in this video, let's discuss about cell membrane. Okay. So, the first topic which we are going to start is the composition of the cell membrane. Now, you can clearly see here the cell membrane, the bilipid layer. Now, what actually the cell membrane is made up of? Okay, we know all the cells are having the cell membrane. Now, this cell membrane is made up of what? Is it made up of proteins, carbohydrates, or lipids, or all three together? Actually, yes. The cell membrane, it is made up of composition of cell membrane is proteins followed by lipids and lastly carbohydrates. Okay, so proteins, lipids and carbohydrates. In this cell membrane, whatever you are looking at, it is having all the three components: proteins, carbohydrates as well as lipids. Here I have written everything in an order. Proteins are there, but what is the composition of this protein? What is the percentage of these proteins? See, in the cell membrane, the majority component, the major component is the proteins. Almost 55%, okay, 55% of the cell membrane is made up of the proteins. So, it is considered as major component. The major composition itself is the proteins. Okay. Now, lipids. When I am talking about the lipids, the composition of lipids is somewhere around 40% and carbohydrate composition is just only 5%. Now, please tell me, what is the minor component in the cell membrane? The minor component in the cell membrane is carbohydrates. Okay. So, after this, they will ask you the exceptions part. You know, everywhere the proteins are the major components. Okay. But what is the exception? See, in Schwann cell membrane, the composition of lipids and proteins, okay. So, the lipids composition is somewhere around 80%. 80% of the Schwann cell membrane is made up of lipids, but proteins are only just 20%. Okay, 20% is proteins and lipids composition is 80%. This is the MCQ. Here itself, I just want to integrate. Guys, are you able to remember anything about the Schwann cell membrane? What are the Schwann cells? Where do we have studied about the Schwann cells? Schwann cells, let me integrate. The Schwann cells, these are myelinating cells. Where? In central nervous system or peripheral nervous system? In peripheral nervous system. So, Schwann cells are the myelinating cells in the peripheral nervous system. That's the cranial nerves as well as the spinal nerves. The cranial nerves and spinal nerves, they are a part of peripheral nervous system. And who is going to myelinate the neurons in peripheral nervous system is the Schwann cell. And what is the importance about the Schwann cell? Schwann cell membrane have highest composition of the lipids but not the proteins. But everywhere in the body, the highest concentration of the cell membrane is proteins only. Okay, that's the 55%. Now, after this, what else you should know for your exams is fluid, mosaic, model. Okay, fluid mosaic model. So, what is this fluid mosaic model? The fluid mosaic model of a cell membrane is given by two persons. One is Singer and Nicholson. Okay, so Singer and Nicholson, these are the persons who have given or who have described the cell membrane in the form of a fluid and mosaics. So, according to them, the cell membrane is just like a fluid. Okay, it's like a fluid, bilipid, like fluid. And in this bilipid sea, the proteins are just floating and they are considered as a mosaics. Okay, so in the sea of bilipid layer, you can clearly see here the blue color things which I am showing you is a bilipid layer. It's a lipid. Now in the sea of lipids, now here and there you can see the proteins, they are just floating. So this is called as fluid and mosaics. According to them, the fluid is the bilipid layer, bilipid layer, and mosaics are the proteins. Okay, so Singer and Nicholson, they gave this fluid mosaic model. Now let me write here, according to them, what is the fluid? Fluid is bilipid layer. Okay, bilipid layer. And what are the mosaics? In the sea of bilipid layer, the proteins and carbohydrates also, they are just floating. Now, even before going further, I want to add one more point here in the area of this exception. See, we have studied the composition. Okay, composition of the cell membrane. The majority of the cell membrane, the composition is proteins. The major component of the cell membrane is proteins. But in which cell membrane highest concentration of proteins are seen? Okay, now let me write here itself. The highest concentration of proteins is present in which cell membrane? Yes, in every cell membrane, proteins are the highest composition, no doubt. But there is this one cell membrane where the protein concentration is the highest, and that area is highest concentration is seen in inner mitochondrial membrane. Okay, so in the inner mitochondrial membrane, we know the mitochondria is having two layers. Okay, outer layer and as well as the inner layer. Okay, outer membrane, inner membrane. The inner mitochondrial membrane have the highest concentration of the proteins. If you don't find this, there is one more option. Inner mitochondrial membrane, or you can also have the presynaptic membrane. Presynaptic membrane. Okay, so even the presynaptic membranes, you know the synapse, the presynaptic membrane and the postsynaptic membrane, in between them the gap is synapse is there. Now in this presynaptic membrane also the highest concentration of proteins are seen, and inner mitochondrial membrane also highest concentration of proteins are seen. The single best thing is the inner mitochondrial membrane. Now after this, let's discuss about the lipids in the cell membrane. The lipids. Let me write here. So now we are going to start with the lipids. Later we will discuss about the proteins and carbohydrates. First, lipids. The lipids in the cell membrane, they are divided into three types. What are they? First type is called as the phospholipids. Okay. Second type of lipids in the cell membrane are called as the glycolipids. And third type of lipids are called as sterols. So, what we have studied that, see, the cell membrane, it is made up of proteins, carbohydrates and lipids. Now, let's begin with the lipid part. So, let's see what are the different types of lipids and what are their importance. And after that, let's discuss about the protein part. Okay, so phospholipids. What are the different types of phospholipids? Now, let's discuss. The different types of phospholipids are, first of all, what is a phospholipid? To a lipid molecule, if a phosphate group is attached, for example, there is this lipid molecule, this is lipid. Now, if phosphate group is attached, then it is called as a phospholipid, right? So, the first type of phospholipid which I want you to know is called as phosphatidylcholine. Okay, phosphatidylcholine. Actually, the real name is, okay, let me write the real name, the full form, real name is in the full form, dipalmityl phosphatidylcholine. Okay, so DPPC, dipalmitoyl phosphatidylcholine. This dipalmitoyl phosphatidylcholine is also called as lecithin. Okay, so this is the first type of phospholipid. The second type of phospholipid is called as sphingomyelin. Okay, the second type of phospholipid is sphingomyelin. The third type of phospholipid is phosphatidylserine. And fourth type is phosphatidyl inositol. And last but not the least, the last type of phospholipid which I want you to know is cardiolipin. Okay, so in your exam they can ask you, please rule out, or they can ask you something like, all of the following are the phospholipids except. So you should know what are the phospholipids. Here I have shown you 5 different types of phospholipids. What are they? Dipalmityl phosphatidylcholine, sphingomyelin, phosphatidyl inositol, phosphatidyl serine, and cardiolipin. These are the 5 different types of phospholipids. Now let's discuss about one by one. Okay, first let's begin with the phosphatidyl dipalmitoyl phosphatidylcholine. Also called as DPPC. This dipalmityl phosphatidylcholine, guys, please tell me, it is also called as lecithin. Okay, now what are the importance? Yes, it's a part of the cell membrane, no doubt. It's a phospholipid, yes, it's a part of the cell membrane, but this dipalmityl phosphatidylcholine is also a major component. Okay, this DPPC, it's a major component in surfactant. Okay, so there is something called as a surfactant. Now in this surfactant, the major component is what? The major component is lecithin or dipalmityl phosphatidylcholine. For your understanding, let me tell you what is the surfactant. See, the surfactant is present, or it is produced by type 2 pneumocytes. Okay, it is produced by type 2 pneumocytes. Now, where these type 2 pneumocytes are present in the alveolus, type 2 pneumocytes in the alveolus. Now, this surfactant, what it will do is, it decreases the surface tension. Okay, it decreases what? It decreases the surface tension. So, in your alveolus, there are cells called as type 2 alveolar cells or type 2 pneumocytes are present. And what they are producing? They are producing a substance called as surfactant. Now, this surfactant, what it is doing? It decreases the surface tension. The point which I want you to know here is, sir, this is surfactant molecule. Okay, this surfactant is there, right? It is made up of what? It is made up of lipids as well as proteins. First of all, let me write here. This surfactant, it's a lipoprotein. So it is made up of both lipids as well as protein. Which lipids, sir? Major lipid and minor lipid are there. Okay, so here listen, the surfactant, it is made up of look here, both lipids as well as proteins. Now, which lipids? There are 2 types of lipids. In the surfactant, there are 2 types of lipids. Major lipids and minor lipid. Okay, major lipids, minor lipids. What is the example of the major lipid? The major lipid which I am discussing here is our dipalmityl dipalmitoyl phosphatidylcholine. So this dipalmitoyl phosphatidylcholine is a major lipid. And what are the minor lipids? What is the minor lipid in the surfactant? The second phospholipid. The second phospholipid which we have discussed. Guys, are you able to recollect this one? The second phospholipid is called as the sphingomyelin. So this sphingomyelin is the minor lipid. Okay, so sphingomyelin is a minor lipid and dipalmitoyl phosphatidylcholine is the major lipid in the surfactant. Okay, done. Now, here, what I want you to know is some integration. If there is a deficiency of surfactant, it will cause which disease? See, surfactant, it decreases surface tension. If there is no surfactant, surface tension in the alveolus will be more. See, remember the surface tension is a collapsing force, it's a destructive force. It collapses everything. So imagine in my body, if surfactant is not there, what happened to the surface tension in my alveolus? Surface tension increases. If surface tension increases, what it will do? It will cause the collapsing of the lungs. Okay, collapsing of the lungs. So the same thing if it happens in a baby. Okay, in a baby, normally surfactant will be produced by 28 to 34 weeks. Okay, but in a baby, if surfactant is not there, what will happen? The baby is going to suffer with respiratory distress. Why? Because the alveolus is collapsed, the lungs are collapsed. Okay, so deficiency of surfactant is going to cause neonatal respiratory distress syndrome, RDS. Okay, respiratory distress syndrome. Okay, neonatal respiratory distress syndrome is also called as hyaline membrane disease. Okay, hyaline membrane disease. So deficiency of surfactant is going to cause NRDS, neonatal respiratory distress syndrome, also called as the hyaline membrane disease. Now, after this, major lipid, both the major lipid and minor lipid of the surfactant are completed. Now, the third phospholipid which we have discussed is what? Phosphatidylserine, right? Before phosphatidylserine, let's discuss about something called as phosphatidylinositol. Okay, this phosphatidylinositol, what are the important points which you should know? Regarding this phosphatidylinositol, we will discuss much in much detail in the topic of cellular messengers or secondary messengers. Okay, now here just believe me and remember this phosphatidylinositol, it acts like a secondary messenger. Okay, phosphatidylinositol first. Okay, it is also called as inositol triphosphate. Inositol triphosphate, IP3. So this inositol triphosphate, this IP3 molecule, it acts like a secondary messenger. Okay, so what it will do, by acting like a secondary messenger, it increases the calcium influx. Okay, it increases the calcium influx into the cells. Because of this calcium, later contractions will happen. Okay, muscle contraction. Okay, is going to happen. So phosphatidylinositol, yes, it's a phospholipid. It's acting like a secondary messenger. This phosphatidylinositol is also called as inositol triphosphate. This IP3DAG, in detail we will study later. This IP3DAG system, inositol triphosphate diacylglycerol system, it will act like a secondary messenger and increases the calcium influx into the cell, which will cause the contraction of the cells. Okay, so done. Now after this, let's discuss about cardiolipin. Okay, cardiolipin. Now what you should know regarding the cardiolipin? See, there are certain diseases in which, or before that, the maximum cardiolipin is present where? Maximum cardiolipin is present in where the maximum cardiolipin is present in the mitochondrial membranes. Okay, maximum cardiolipin is present in the mitochondrial membranes. Remember cardiolipin, is it a phospholipid, is it a glycolipid, or is it a sterol? It's a phospholipid. Okay. Now, the point which I want you to know is, there are certain diseases in which antibodies are produced against this cardiolipin. Okay. So, anti-cardiolipin antibodies are present in which disease? Okay, anti-cardiolipin antibodies, they are present in syphilis, APLA syndrome, antiphospholipid antibody syndrome, APLA syndrome, and lastly SLE, systemic lupus erythematosus. See, there are certain diseases in which the patient will produce antibodies against a phospholipid, and that phospholipid is nothing but the cardiolipin. So, anti-cardiolipin antibodies are seen in conditions like syphilis, APLA syndrome, and SLE. Just trying to integrate. I hope you already studied that if there is a person who is suffering with SLE, what the patient is going to have? The one thing which every time comes into the mind when I am talking about the disease called as SLE, the patient is going to have what? Which rash? It's a malar rash. Okay? Or malar rash. Okay? So there will be a butterfly shaped rash which is present on the malar region, malar eminences. It's the malar rash is going to be present. And APLA syndrome, remember APLA syndrome means antiphospholipid antibody syndrome. And what is that antiphospholipid? What is that phospholipid? Cardiolipin. Okay. See, if a person suffering with the APLA syndrome, especially females, this question will come in obstetrics. Okay. APLA syndrome, if a female is suffering with APLA syndrome, that she is going to suffer with recurrent abortions. Okay, then she is going to suffer with the recurrent abortions. Okay, because of the placental insufficiency. If a patient is suffering with the APLA syndrome, clots are going to form within the placenta, which will cause the impairment in the oxygenation for the baby, and that will lead to the abortions. That's a total different thing. I'm not going in that detail. Recurrent abortions. APLA syndrome is going to cause a recurrent abortions, and this recurrent abortions are going to be seen when? Is going to be seen during second trimester. Okay, so APLA syndrome is going to cause second trimester recurrent abortions. Done. And syphilis. 2-3 important points about the syphilis. Syphilis is a sexually transmitted disease, is a STD. It is caused by which organism? It is caused by an organism called as Treponema pallidum. Okay, Treponema pallidum. And this syphilis is caused by this, Treponema pallidum. And this Treponema pallidum, one question will be repeatedly asked in your exams. It will be tested in your exams. It's a spirochete. Okay, it's a spirochete or spirochete. So it's a spiral in shape. These organisms are spiral in shape. Okay, that's why they are called as the spirochetes. Do you know how they will move? If you look at their motility, they will move like a corkscrew. That's the MCQ. So this Treponema pallidum, it have corkscrew motility. Okay, it have the corkscrew motility. And what is the test that is done? What is the screening test that is also repeatedly tested in the exams? What is the test that is done for this syphilis? And that test, there are many tests, but in your exam, they will ask you, it is screening test. There is a VDRL, Venereal Disease Research Laboratory test, VDRL test. Okay, it is done for the syphilis. So, just integrations part is also done. Now, what I was discussing? Cardiolipin. Anti-cardiolipin antibodies are seen in which condition? Anti-cardiolipin antibodies are seen in 3 conditions: SLE, Apla syndrome, as well as syphilis. Now, after this, what you should know is the last phospholipid. The last phospholipid is phosphatidylserine. Okay. Now, what do you need to know about the phosphatidylserine? Guys, please look here in a healthy cell. Okay, look here. In a healthy cell, this phosphatidylserine molecules, all these molecules, okay, all these cone-shaped molecules, these are the phosphatidylserine molecules. Where they are present? The phosphatidylserine molecules, they are present towards the cytoplasm. This is the side of cytoplasm. Okay, they are towards inside the cell. They are internalized. Okay, this is the outside of the cell. This is the inside of the cell. Normally, phosphatidylserine molecules are present towards the cytoplasm. They are not exposed out. But the point which I want you to know is, look at here. What is this cell? This is a cell which was about to die. So, it's undergoing what? It's undergoing apoptosis. But in an apoptosing cell, something important happens. See, these phosphatidylserine molecules, these PS molecules, now they are getting externalized. Okay, now what they are getting? They are getting externalized. Whenever they are getting externalized, what happens is, if you want to know whether a cell, which cell is undergoing, you are having a group of cells, okay, you are having a bunch of cells. Now, in this group of cells, which cell is undergoing apoptosis? How can you know? By using a dye or by using a marker called as annexin. So, annexin 5 is a marker. Okay, these molecules which we are showing here, these blue color molecules, these are the annexin 5 molecules. Normally in a healthy cell, this annexin 5, okay, is it able to bind with the phosphatidylserine? No. This annexin 5 molecules, they will come, but they cannot fix with the phosphatidylserine. But in a dying cell or in an apoptosing cell, as the phosphatidylserine molecules are externalized, now this annexin 5 can go and bind with the phosphatidylserine. Now, whenever this annexin 5 binds with phosphatidylserine, it will give fluorescence. So that's why you can see the fluorescence is coming. It's giving the light. It's emitting the light. So that's why an apoptotic cell will give the fluorescence. Okay, see, bright fluorescent signal will be given because of this reagent, that is the annexin 5. So the point which I want you to know is, in a healthy cell, first point, in a healthy cell, phosphatidylserine is internalized. Okay, in a apoptosing cell, phosphatidylserine molecules, they are externalized. Okay, they are externalized. Next, what is the marker of apoptosis? Marker of apoptosis is annexin 5. Okay, annexin 5 is considered as the marker of apoptosis. Okay, so with this we have completed the phospholipid parts. Okay, we have discussed about the different types of phospholipids. Now, after these phospholipids, now let's go back. Yeah, look here. So, phospholipids are completed. Dipalmityl phosphatidylcholine, sphingomyelin, phosphatidylserine, the last one which we have discussed, phosphatidylserine, phosphatidylinositol, the secondary messenger, and the cardiolipin. All these are completed. Okay, all these phospholipids are completed. Now, after the phospholipids, now let's discuss about the glycolipids. Now, how many types of glycolipids are there? The glycolipids are of two types: cerebrosides gangliosides. Okay, cerebrosides are there and gangliosides are there. Now, here, what do you need to know for your exams regarding the gangliosides? There are different types of gangliosides, like GM1 ganglioside, GM2 ganglioside. There are different types of gangliosides. Now, especially what you need to know is regarding the GM1 ganglioside. So, what is this GM1 ganglioside and where it is present? This GM1 ganglioside, yes, it's a lipid, glycolipid. Where it is present? It is present in the intestinal cells, or it's present in the enterocytes. Now, imagine this is one enterocyte. Okay. Now, on this enterocyte, okay, this is the intestinal cell, enterocyte. Now, on this intestinal cell, there is this glycolipid present. Okay. Now, this glycolipid which I am showing you is the GM1 ganglioside. Okay. Now, this is GM1. Now, in an infection called as cholera, everyone knows this cholera is because of what? Cholera is because of Vibrio cholera. It's a bacterium, right? Now, this Vibrio cholera, okay, this bacteria, it is going to produce a toxin. Okay, so cholera toxin. Cholera toxin. See this cholera toxin, what it will do, this cholera toxin, it is using this GM1 ganglioside. Okay, it is using this glycolipid, this receptor. Okay, so this glycolipid is acting like a receptor for the cholera toxin. So this cholera toxin, now it will enter into the intestinal epithelial cells. This is the toxin. Okay. Now when the toxin enters into this epithelials, enterocytes, now what this toxin will do? This toxin, it is going to activate the G proteins, the G protein complex, like which will activate the secondary messenger system. So this toxin is going to activate the G protein system. Now when the G protein system is activated, at the end of the day, okay, the G protein adenylyl cyclase will be activated, because once the adenylyl cyclase is activated, it's going to increase the cAMP levels. The cAMP levels in the cells are going to be increased. When the cAMP levels are increased, at the end of the day, let me tell you what happens is, now more and more water, sodium, chloride and water, wherever sodium goes, water follows. So sodium, chloride and water, they are more and more secreted into the GIT. Okay, they are more and more secreted into the GIT. So more water is being secreted into the GIT, that will cause diarrhea. So the cholera patient is going to have diarrhea. Okay, the patient is going to have the diarrhea, and this diarrhea is typically called as rice watery diarrhea. Okay, so the patient is going to have the rice watery diarrhea. So now tell me, what is that important point which I want you to know is, sir, glycolipids are of two types, erythrocytes as well as the gangliosides. Ganglioside, the important ganglioside is the GM1 ganglioside, which is present on the intestines or the enterocytes. Now this GM1 ganglioside, it acts like a receptor. It acts like a receptor for the cholera toxin. So cholera toxin enters into the cell, enterocytes via the GM1 ganglioside. This cholera toxin activates the G protein complex and further it activates the adenylyl cyclase, so that the intracellular cAMP levels increases, because of which more and more sodium, chloride and water are being secreted into the GIT, lumen, the lumen of the GIT, that will cause diarrhea. So we all know, right, cholera patients are going to suffer with the diarrhea, rice watery diarrhea. So this is how the patient is going to have the diarrhea. This is the mechanism of action. Now here itself, as an integration part, I want you to know, if the person is suffering with typhoid, okay, if the person is suffering with typhoid, then also the patient will have diarrhea. Typhoid is an enteric fever, right? It's an enteric fever, the patient is going to have diarrhea. Okay, but what kind of diarrhea? It is called as pea soup diarrhea. Okay, the patient is going to have the pea soup diarrhea. Cholera will cause rice watery diarrhea. Typhoid will cause pea soup diarrhea. Okay, this is the point which I want you to know. So with this, glycolipids are also completed. We have completed the glycolipids also. Now after this, let's discuss about the proteins. Okay. So lipids are completed. Now we are discussing about the proteins. How many types of proteins are there? The proteins in the cell membrane, they are divided into three types again. First type is called as the transmembrane proteins. Okay, transmembrane protein are the integral proteins. The second type are called as the peripheral proteins. And the third type of proteins are called as the lipid anchored proteins. Okay. So there are many types of proteins, but majorly I want you to know. So proteins in the cell membrane are transmembrane proteins, peripheral proteins, and lipid anchored proteins. What are the differences between these? See, transmembrane protein means, for example, look here. Transmembrane protein means, for example, if this is a cell membrane, this protein molecule, it is present outside the cell, it is passing through the cell, through the cell membrane, and it is present even inside the cell. Okay, so this is like traversing, it's like a passing from outside to inside, and it is present throughout the cell membrane. For example, you know about this receptor called as the 7-pass receptor or the serpentine receptors. So this serpentine receptor, it's a protein, it's an example of transmembrane protein. Why? Because it's there outside the cell, it's having the intracellular domain, extracellular domain, and it's passing throughout the cell membrane. So this is an example of transmembrane proteins. And even, for example, you know about the channels. If this is a cell membrane, okay, this is a cell membrane. Now there are certain channels. Now these channels are allowing the substances to pass in and out of the cell. So these channels, like sodium channel, chloride channels, they are also examples of, okay, sodium, potassium, ATPases, all these channels, sodium channels, potassium channels, chloride channels, sodium, potassium, ATPases, the pumps, all these are examples of, again, the transmembrane proteins. Then what are the examples of peripheral proteins, sir? Okay, first let me write the examples. The examples of the transmembrane proteins are, for example, receptors. Which receptor? The classical example is 7-pass receptor. Or the serpentine receptor, first example. The second example which I want you to know is the pumps. Which pumps? Sodium, potassium, ATPase. Sodium, potassium, ATPase. Okay, sodium, potassium, ATPase. It's a pump, it uses the energy. And third example which I want you to know is channels. Okay, which channels? Ion channels. Okay, the ion channels, the ions which are moving sodium chloride, these ion channels. So these are the examples of the transmembrane proteins. Now what are the examples of the peripheral proteins? First of all, what is a peripheral protein? Means, look here, this is the cell membrane. Okay, this is here I am showing you the cell membrane, right? Now either the protein is present outside or inside. For example, see, the protein is present inside. Okay, it's a cell membrane protein only, but it's present on either one side, out or in. So this is an example of this is an example of peripheral protein. Now what are the examples of peripheral proteins? Here, this is a very important area. The examples are 1. Spectrin, ankyrin, and third example is dystrophin. Okay, so spectrin, ankyrin, and dystrophin, these are the peripheral proteins. Now, in exam point of view, they will ask you, see, RBC, okay, the spectrin and ankyrin, especially they are present in the cell membrane of the RBC. Okay, and what is the importance? They maintain the biconcave shape of the RBC. We know that RBC is having which shape? RBCs are having the biconcave shape. Okay, biconcave shape. This biconcave shape is because of this peripheral proteins like spectrin and ankyrin. Now if there is any defect in spectrin defect, okay, that will cause definitely a change in the RBC shape. So defect in spectrin will cause which disease? It will cause hereditary elliptocytosis. Okay, hereditary ellipse, like elliptical shape. Okay, so hereditary elliptocytosis is because of the defect in the spectrin, and defect in ankyrin will cause hereditary spherocytosis. Okay, spheres means like round shaped RBCs. RBCs are going to be round in shape. Okay, now this dystrophin. Sir, where is this dystrophin present? Dystrophin, it is present in the muscles. Okay, again it's a peripheral protein which is present in the muscles. It is present in the muscles. Now, dystrophin gene, some important points. Dystrophin gene is the largest gene in the humans. Dystrophin is a protein. A protein will have a gene, right? So, the dystrophin gene is the largest gene in human genome. Okay, so I will discuss about this dystrophin in detail in the muscle also. Okay, in the part of muscle contraction, how muscle contracts, what is dystrophin, and why it is needed, how it gives the tensile strength to the cell membrane. That we will discuss later. But here, the point which I want you to know is dystrophin. It is a peripheral protein. Dystrophin gene is the largest gene in the human genome. Okay, what happens if this dystrophin is defective? If the dystrophin, if it is defective, okay. If the dystrophin is defective, then it is going to cause a condition called as Duchenne's muscular dystrophy. Okay, simply called as a DMD, Duchenne's muscular dystrophy. Okay, so this Duchenne's muscular dystrophy, it is due to absence. Okay, so it's complete absence. If this dystrophin, if this protein is completely absent, then it is going to cause a disease called as Duchenne's muscular dystrophy. Now, if this dystrophin, if it is a defective, okay, so it's defective, not complete absence, defective, then it is going to cause a disease called as Becker's muscular dystrophy. Okay, see, Becker's is relatively a milder condition. Becker's is more severe condition. Duchenne's is more severe conditions. Becker's is a milder condition. Okay, now in many of the exams, this question has been asked. In Duchenne's muscular dystrophy, okay, if I am the person, imagine if I am the person who is having the Duchenne's muscular dystrophy, the dystrophin is not there. Which protein is defective? Dystrophin. Okay, and I shouldn't say defective. It is absent. The dystrophin is absent. Now, when the dystrophin is absent, do you think the muscles are going to have a proper strength? No, the muscles are not going to work normally. So, there is muscle weakness. The patient is going to have muscle weakness and the muscles are getting atrophied. Okay, so the patient is going to have atrophy of muscles that lead to muscle weakness. And especially in this patients who are suffering with the Duchenne's muscular dystrophy. Okay, imagine there is a small boy, a 10-year-old boy. He is suffering with the DMD, Duchenne's muscular dystrophy. So if he squats, if he is sitting down, and if you ask him to stand up, okay, if you ask him to squat and ask him to stand up. Now a normal person without using the upper limbs, he can simply stand up like this. But this person who is suffering with the Duchenne's muscular dystrophy, DMD patient, he cannot lift his body just with his legs, just by using his legs he cannot stand. Why? Because the muscles in his legs and thighs, they are weak. The dystrophin is not there. So he will use the support, okay, he will use the support of even the upper extremities to lift his body. Okay, the sign is called as Gower sign. This was the question asked in the last FMGE exam. So, Gower sign. Okay, Gower sign is seen. Gower sign is going to be positive in Duchenne's muscular dystrophy. Okay, in the DMD Duchenne's muscular dystrophy, Gower sign is positive, where the fellow is using all his 4 extremities, all upper limbs and lower limbs to lift his body up. Gower sign. And as integration part, here itself I am telling you, the muscles are getting atrophied, especially in many places of the body, especially in the legs, in the calf region. The muscles are totally atrophied, and in the place of the muscles, the fat is getting deposited. So if you look at the calves are going to be looking bigger in size. You will think that, oh my god, calf hypertrophy is happening. This is calf hypertrophy, but this is not a calf hypertrophy, but rather it's a adipose tissue. Adipose tissue is getting deposited over there. The muscles are getting atrophied. So that's why this is called as pseudo calf hypertrophy. Okay, the patient is going to have what? Pseudo calf hypertrophy. If you look at his calves, they are going to be bulkier in size. Pseudo calf hypertrophy. And in this patient, Duchenne's muscular dystrophy, the patient is having weaker muscles. So, what is the most common cause of death in these patients? It's a respiratory failure. Diaphragm is also a skeletal muscle. Okay, it's also a skeletal muscle, right? So, as the dystrophin is not present, this diaphragm cannot contract properly. That will lead to respiratory failure. So, cause of death is respiratory failure. Because diaphragm is also a skeletal muscle. Okay, diaphragm is also a skeletal muscle. So diaphragm do not contract properly. So that will cause respiratory failure. So with this, we have discussed about the spectrin, ankyrin as well as dystrophin. Now after this, what we are left with? We are left with which type of proteins? The lipid anchored proteins. Now we will discuss about the lipid anchored proteins. Now, look here guys. What is this cell? This cell is the RBC. Okay. Now, on this cell, there is this one protein present which is attached to the bilipid layer. Okay, it is attached to lipids. So this protein which I am showing you is called as a GPI. Okay, GPI. So this GPI molecule, where it is present in the cell membrane. This GPI is an example of lipid anchored protein. Okay, it's an example of lipid anchored protein. It attaches to a lipid molecule. Okay, it attaches to the phospholipid molecules. Okay. Now, what is the importance of this is, see, there is a condition called as, okay, paroxysmal nocturnal hemoglobinuria. Okay, so PNH, simply in a short form we will call it as PNH. Okay, paroxysmal nocturnal hemoglobinuria. So what is this condition? Paroxysmal nocturnal hemoglobinuria. What is happening? Guys, let me tell you something. Now in our blood vessel, okay, imagine this is the blood vessel. This is a little conceptual thing. This is your blood vessel, right? Now in your blood vessel, which cells are there? RBC are there. Okay, RBC are there. WBC are there. Okay, RBC, WBC, and small platelet molecules are also there. RBC, WBC, small platelets are also there. Okay. Now imagine if you are suffering with an infection, there is this microbe entered into your body. Okay, this microbe entered into your body. When the microbes are entered, definitely complement proteins in your blood are going to be activated. Okay, so this bacteria, it is going to activate, you know about the classical pathway. In pathology we will study, there is something called as a classical pathway, okay, which will ultimately activate certain proteins called as a clotting proteins. So not clotting, complement. Okay, complement proteins. So whenever you are suffering with an infection, the complement proteins C3, C4, C5, C6, C7, some complement proteins are there in your plasma along with the RBC, WBC platelets. In your blood, complement proteins are also there, but normally complement proteins are in inactive state. But whenever you get infection, these complement proteins are activated. Now these complement proteins, what they will form? These activated complements, they will form something called as a membrane attack complex, MAC. This membrane attack complex, it is going to attack, it is going to fall onto this bacteria. Now it will kill the bacteria, for sure, no doubt. But my point is, these complements are like blind soldiers. These complements, they are like a blind soldiers. Once they are activated, they are going to start firing. Yes, they will kill the bacteria, but there is a possibility that this membrane attack complex, this activated complements, they can also attack your platelets, they can also attack your RBC, and they can also attack your WBC. Am I right or not? They are blind, they don't know what is self and what is foreign. Okay, once if membrane attack complex is activated, yes, they will start to attack both the bacteria as well as your RBC, WBC and your platelets, your blood cells. Okay, so what we want, do you want RBC destruction, WBC destruction, platelet destruction? No, we don't want that. Okay, we don't want that. So that's the reason why nature have made our cells, nature given a defense. Do you know what is this defense? The defense is this GPI molecules. See, this lipid anchored protein called as GPI, it is attached with, okay, so to this GPI, two more molecules are attached. They are DAF, okay, DAF means decay accelerating factor, okay, the cluster designation is CD55. So CD55 is DAF, and one more molecule is attached, that is MIRL, MIRL, that is CD59, okay, MIRL. So DAF molecules and MIRL molecules, DAF and MIRL molecules, where they are present? See, this DAF and MIRL, they are present on the GPI. Okay, they are attached to the GPI. Do you know what this DAF and MIRL will do? So this DAF and MIRL, the decay accelerating factor and MIRL, they will inactivate. Let me write here, the DAF and MIRL, what they will do? They will inactivate activated complements. Okay, they will inactivate the activated complements. See, now whenever there is a bacteria, complement proteins are going to be activated. When the complement proteins are activated, membrane attack complex is going to be formed. This membrane attack complex, see, now it is coming onto the RBC. It is trying to attack the RBC. Are the activated complements, they are coming and falling on the RBC. No problem for the RBC. No problem for WBC and platelets. Why? Because on the surface of RBC, DAF and MIRL are present. This DAF and MIRL, decay accelerating factor and membrane induced reactive-lysis, these molecules will inactivate the activated complements. So the activated complements cannot destroy RBC, WBC or platelets. Rather, the activated complements or the membrane attack complex will only attack the bacteria. Okay. Now, let's discuss what happens in a condition called as paroxysmal nocturnal hemoglobinuria. Okay. In paroxysmal nocturnal hemoglobinuria, what happens is, so the patient is having, okay, in PNH condition, there is absence of GPI. Okay, this lipid anchor protein is not there. Whenever the lipid anchored protein is not there, do you think DAF and MIRL are present? So DAF not there, MIRL is also not there. So now your RBC, WBC and platelets, now I am showing you here only RBC. Okay, this RBC is not having any protection, no DAF, no MIRL. Okay, so what happens? Now let's talk about what happens during sleeping. Okay, see, in a simple way you can understand like this. Actually the mechanism is a little different, but for easy understanding you can have this. So sleeping, during sleeping, what kind of breathing you are going to have? So very subtle, shallow breathing is going to be there. Okay, the person is going to have the shallow breathing. So no deep breathing, only shallow breathing. So whenever there is a shallow breathing, what happened to the carbon dioxide? The patient is not like, no, the person is not having deep breathing, only just shallow breathing. So do you think carbon dioxide is going to wash out? No. So during sleeping, usually in you and me, so the carbon dioxide retention will happen. Okay, carbon dioxide is not getting puffed out properly, washed out. So carbon dioxide retention will happen in the lungs. The carbon dioxide reacts with the water in the lungs. So it will form the carbonic acid, H2CO3. So usually during night or during sleeping, the acid production is there, carbonic acid production is there. So usually there is acidosis during sleeping. Respiratory acidosis. This is called as a respiratory acidosis. Because of the respiratory pattern, there is respiratory acidosis. Now, it was believed that even acidosis, okay, even this respiratory acidosis will activate the complements. So during night, the complements in your blood are activated. Okay, but in your body, my body, no problem. Okay, let the complements be activated. If there is any bacteria, that will be killed. But in this person who is having paroxysmal nocturnal hemoglobinuria, now his RBCs are not having protection. Now these activated complements, now they will directly start to attack the RBCs, WBCs and platelets. So during night, what is happening? The RBCs are getting attacked. Okay, the RBCs, the poor RBCs are being attacked. So what will happen to the RBCs? Now the RBC molecules, they are lysed or they are damaged. So what is happening? There is hemolysis. There is hemolysis during night, sir. Okay, there is hemolysis. So whenever the RBCs are getting broken down in the plasma, RBCs are supposed to be broken down in the spleen. That's the graveyard. But when the RBCs are getting broken down in the plasma, what happens? That will cause leakage of the hemoglobin. Okay, the hemoglobin molecules are being leaked. Now where this hemoglobin will go? The hemoglobin will come out in the urine. Okay, hemoglobin is filtered and the hemoglobin is coming out in the urine. So this is called as hemoglobin urea. Okay, hemoglobin urea. Hemoglobin is coming out in the urine. So that's why this patient see the name also paroxysmally means sometimes. Okay, nocturnal, when the person is going, like night time hemolysis is happening, and when this person is going to the urine, there is hemoglobin urea, red color urine is there. So this is paroxysmal nocturnal hemoglobin urea. Okay, why all this problem? It is because of the absence of a lipid anchored protein called as GPI. Whenever this lipid anchored protein is not there, DAF and MIL, decay accelerating factor and membrane induced reactive lysis, they are not present. Whenever they are not present, what happens? RBCs are not having any protection from the activated complements. The activated complements during night will attack the RBC, leading to hemoglobin urea. See, it's a problem but not a life threatening. But the life threatening problem for this patient is not the hemolysis, but during night what happens? These activated complements are not only attacking the RBC, but also the platelets. So the platelets, thrombolysis, the platelets are also getting lysed away. So what that will cause? That will cause clotting. Okay. So in these patients, the most common cause of death is thrombosis. Okay. Why? Because platelets are also getting broken down. The rupture of the platelets, the broken down of the platelets will trigger the clotting cascade. That clotting cascade can cause the intravascular clots leading to thrombosis, and that can actually kill the patient. So these are the points which I want you to know. So now tell me, how many types of proteins are there? There are 3 types of proteins: the transmembrane proteins, the proteins which are going throughout the membrane, transmembrane proteins, and there are proteins like peripheral proteins, pectin, ankyrin and dystrophin. Pectin defect will cause hereditary electrocytosis. Ankyrin defect will cause hereditary spherocytosis, and dystrophin defect will cause Duchenne's muscular dystrophy. And lipid anchored protein, the example of a lipid anchored protein, the protein which is attached to the lipids, okay, lipid components. See, such type of proteins are called as the lipid anchored proteins. Lipid anchored protein is the GPI. The defect of the GPI is going to cause a condition called as paroxysmal nocturnal hemoglobin urea. So with this, the part 1 is done. Now let's have one more part in the topic of the cell membrane and see some more important MCQs. Okay, mainly in the MCQ point of view, and let's try to cover some more important clinical integrations. Okay guys, hope the video is helpful. Thank you.